Hello, I'm Jason Bobe, an associate professor in the Department of Genetic and Genomic Sciences at the Icahn School of Medicine at Mount Sinai. Today, I'm going to highlight some key progress over the past 5 years in our understanding of Lyme disease and some gaps that remain. I'll cover four main areas: prevention, fundamental knowledge, clinical and translational medicine, and finally, community building. I'd like to let you know that I have no commercial financial conflicts of interest to declare. First, prevention. For ecological prevention, I'd like to highlight the Tick Project. They have undertaken a massive randomized controlled trial of ecological interventions to prevent Lyme disease. They are comparing the effectiveness of a fungal spray and rodent bait boxes across 24 neighborhoods in Dutchess County, New York. To my knowledge, it is the first ever randomized, placebo-controlled, double-blinded study of the ecological prevention of tick-borne disease. The much anticipated results are due early next year. Next, human vaccines. It has been 20 years since we've had a licensed Lyme vaccine for people. I'm happy to say there is now a new vaccine in human trials from a company, Valneva. This is a next-generation OSPA vaccine with epitopes from multiple pathogenic species represented. And there are also multiple other promising vaccine candidates under development in preclinical stages. Second, fundamental knowledge. Investments in basic science are hugely important to progress in tick-borne disease. Currently, around 1,000 scientific papers are published each year that mention the term Lyme disease. Here are some highlights from a few of these. In 1982, the Lyme disease agent was identified, Borrelia burgdorferi. By 2010, the Borrelia burgdorferi species complex had expanded to 18 species. Since 2010, there has been continued growth in the number of new Borrelia species identified from patient samples, ticks, or vertebrate hosts, such as the new species identified in 2016 in the upper Midwest called Borrelia mayonii. Remarkably, this was identified by screening over 100,000 patient samples. Somebody noticed something unusual among a handful of those samples, and the discovery soon followed. Many advances in our fundamental knowledge of Lyme disease have been aided by the development of better technologies, such as affordable DNA sequencing. Dozens of Borrelia species and strains have now been sequenced. As Lyme invades Canada, our neighbors to the north have swung into action. Researchers there recently contributed 64 whole genomes of Borrelia isolates collected from ticks. Another group of researchers took many of the public Borrelia genomes and compared them. They found these spirochetes could be divided into two clades or groups. So the genus Borrelia has been split into two. The genus Borrelia now denotes relapsing fever agents and their relatives, and the genus Borreliella denotes Lyme disease agents and their relatives. To help with pronunciation, I'm joined by Alan Barber.
The new name for the genus for the Lyme disease agents is Borreliella, which means Borrelia-like. Borreliella.
The scientific community has examined the microbial DNA inside of ticks collected from endemic regions to assess their pathogen burden. In New York and Connecticut, the data is sobering. The pathogen burden in ticks can be astoundingly high. Around 60% of ticks in these regions carry Borreliella burgdorferi, or simply BB, and some ticks carry more than one human pathogen. The black-legged tick, Ixodes scapularis, has also had its genome sequenced. With all of these genomes, we're in a better position to understand how these stealthy biters and their cargo are put together. And from this knowledge, we may find routes to new interventions. What molecules do ticks produce that help them mask pain and itch from their bites? Can these be blocked? How does Borreliella evade the human immune system? And how might we short-circuit those defenses? By studying the biology of these spirochetes, we've also learned about other unique characteristics. Virtually all bacteria have peptidoglycan, a structural molecule in their cell walls. It turns out the peptidoglycan in BB is chemically unique and unlike any previously studied. As these spirochetes grow, they shed these peptidoglycan fragments. If you inject this molecule into the tail of a mouse, it gets arthritis. Could this molecule be a source of ongoing inflammation in patients? More research is needed, but this is a promising finding. Third, clinical and translational medicine. The Lyme community knows how challenging getting a proper diagnosis can be for some patients. Without better diagnostics, patients may receive inadequate or improper treatment. What everyone hopes for is a highly sensitive and specific diagnostic test for all pathogenic species that also works at all stages of Lyme disease. While I'd love to say there's a breakthrough diagnostic that has been carefully validated in well-characterized patient cohorts, we're not there yet. However, dozens of promising diagnostics are in the pipeline, and existing diagnostic tests continue to be scrutinized and refined. In order to effectively treat patients with persistent Lyme disease, we need to better understand the underlying pathophysiology of the disease. We need patients working hand in hand with the scientific research community to do this. The longitudinal and cross-sectional slice studies based at the Johns Hopkins Lyme Disease Research Center have shown that patients with persistent Lyme can be recruited into research and retained at high levels over many years. Through the slice studies, there has been a rapid accumulation of high-quality clinical and laboratory evidence on large numbers of patients. Serology, immune repertoire profiling, levels of cytokines and other immune mediators, RNA sequencing, DNA samples, etc. Patients are also administered validated instruments for the measurement of symptoms, such as cognitive function, or the impact of the disease on their quality of life, among others. Some important biomarkers for different disease states have been identified from these and other studies. C-reactive protein and CCL19 levels in US-based patients with persistent Lyme, levels of IL-23 among European patients with persistent Lyme, and for antibiotic refractory Lyme arthritis patients, levels of gamma interferon. Many significant gaps remain. Diagnosis of persistent Lyme lacks an objective laboratory test. Instead, diagnosis is achieved through a process of exclusion. And the recovery from persistent Lyme also lacks an objective test. Currently, there are no prognostic indicators to stratify patients for susceptibility to persistent Lyme. And there are no FDA-approved curative therapies for persistent Lyme. Safety and efficacy data for off-label treatment regimens and complementary therapies for persistent Lyme are not well established. There remain many unanswered questions about the pathophysiology of persistent Lyme. Is susceptibility to or protection from persistent Lyme a function of the patient or the infecting pathogen or pathogens? What are the underlying causes of ongoing inflammation and other health problems some people experience following treatment? Persistent infection or persistent antigenic debris? Immune dysregulation, such as autoimmunity or autoinflammation? Neuronal dysregulation? More research is needed to find the answers. There are multiple labs working on new therapies for Lyme disease. For example, labs at Tulane and Johns Hopkins identified several triple drug combinations that showed efficacy against persistent Borrelia in mice. 2 of these combinations will soon be tested for efficacy in primates. These are oral FDA-approved antibiotics. And I'd like to take a moment to acknowledge the passing of the scientist and drug developer, Dr. Neil Spector. His personal encounter with Lyme disease permanently changed his life. His illness led to a heart transplant and a career change from cancer research to Lyme disease research. His devotion to patients and researchers in the field will be greatly missed. His colleagues at Duke are carrying forward his important work for novel therapies for Lyme disease. Finally, field building. For Lyme disease research to really thrive, we need to attract great researchers, find new ways to collaborate with patients, develop shared scientific resources, and of course, have money to do great work. We've made great strides in some, but not all of these. There are now multiple Lyme disease-related cohorts and a biobank, the Lyme Disease Biobank, the SLICE studies at Johns Hopkins, and the outdoor worker cohort at Stony Brook and Long Island. There are also many passionate researchers who are devoting their lives to solving Lyme disease. You may read about some of these and their work on the new LymeMIND website. Even though Lyme disease is the most common vector-borne disease in the United States, federal funding has been sorely inadequate for many years. So we're all thankful that the Stephen and Alexandra Cohen Foundation has invested tens of millions of dollars towards Lyme disease research in recent years, alongside other disease foundations and philanthropists in the community. Thankfully, government is getting more involved, it seems as well. HHS recently convened a working group on tick-borne disease, and the NIH has announced a strategic initiative in tick-borne disease that hopefully will lead to more funding opportunities and more great science by researchers aiming to help solve Lyme disease. Thank you for your time today.